Hello, everyone. Welcome to the last CGIL seminar of the semester and of the year. Today, we're delighted to welcome Dr. Troy Rowan, who is an assistant professor at the University of Tennessee's Genomics Center for the Advancement of Agriculture. He holds dual appointments in the Department of Animal Science and in the College of Veterinary Medicine's Large Animal Clinical Sciences Department. His research uses genomic and computational approaches to understand the biology that underlies a wide range of complex traits in beef cattle. He is particularly interested in local adaptation, heterosis, novel phenotype creation, and genomic approaches to increasing beef cattle sustainability. His work aims to deliver tangible solutions to US beef cattle producers while answering fundamental biological questions. Additionally, Dr. Rowan serves as the State Extension Beef Cattle Genetics Specialist, providing producer education and outreach in Tennessee and nationwide. And with that, I'll pass it over to you, Troy.
All right, I appreciate the invitation and the introduction, Colin. I'm excited to be with a group here virtually that I really have admired from afar for a really long time. There's tons of really cool work that has and continues to be done up in Guelph. So excited to share some of the work that I'm wrapping up from my PhD work, and then some of the new avenues that my lab here at the University of Tennessee have taken on over the last 18 or so months that I've been here. So with that, I guess the sort of thing that ties all of these what seem like kind of out there pieces together is unconventional phenotype. So hopefully that makes sense by the end of all this. But our big interest is understanding complex traits, understanding selection, and understanding some of these biological phenomenon that really go into livestock breeding programs. And can we harness some of this using unconventional phenotypes, whether that be in the form of sort of non-actually heritable phenotypes or intermediate molecular phenotypes or whatever have you. So that's the thing that really ties together what we're going to discuss today. But again, I'm really excited to be here and share some of this stuff with y'all. So just a little bit about our group here at the University of Tennessee. We, I think, approach things pretty comprehensively, really focused on the beef cow. Our situation here in the state of Tennessee is very forage based. We're great at growing grass, tons of rainfall, no other serious environmental stressors aside from heat and infected endophyte fescue, but we're really a great place to for the cow-calf industry. And so all of our stuff really focuses on ideas of cow efficiency and how we maximize the efficiency of beef production, our number one agricultural commodity here in the state. But from a research perspective, we're really trying to tie together functional genomics, quantitative genetics, and high throughput phenotyping, and stitch that together into some usable form for animal breeders. I don't ever really claim to be an animal breeder myself. I'm somewhere between an animal breeder and a functional geneticist, but I think that there's a need for some crosstalk between these disciplines, with the ultimate goal, of course, being producer application and adoption of these technologies that we're able to deliver on when we combine these things together. Like Colin said, I'm a state extension specialist, so I spent all week out talking with producers and our extension agents that are scattered throughout the state. So really excited about the prospect of delivering this stuff straight to the end user, something that again, the beef industry is especially equipped for. We've got thousands and thousands of independent companies, I guess, in the form of farmers and ranchers that are making their own selection decisions. So it's a unique challenge, but one that I think there's tons of potential, and it's really what gets me up and keeps me going in the morning. And the one thing I wanted to point out, I put a little map here because the fun fact that people started telling me the first week that I got here is that Tennessee, from this little corner here, which is called Mountain City, the farthest east part of Tennessee, is actually closer probably to Guelph than it actually is to Memphis. So Tennessee is a big, long state with lots of sort of geographic diversity and production styles for cattle. So I think it represents a unique system for us to do some of this work. But with that, I'm going to dive right into sort of the big questions that our lab is asking, and sort of how I'll frame my talk today. So the first is, how does selection work to change the genome over time. And this is something that I've been interested in for a while now. And I think it's something that we maybe don't think a lot about in the context of a breeding program is how is the selection that we're doing actually altering that underlying genome, right? We're changing allele frequencies, but of what? And if we find ways to answer those questions, how can that inform the decisions that we make or how we sort of evaluate our breeding programs? The second is, how does heterosis work? I'm also, like I said, very interested in breeding more efficient cows. How do we measure forage-based efficiency in some of these extensive beef cattle operations? And that's something that we're interested in using some novel methods to do, and I'll get to that at the end. And finally, how might we use omics technologies, right? Whether that's gene expression or metabolite abundance or whatever, for phenotyping and eventually entering those things into breeding programs in a logical way. So I'll touch on all these things, but I want to start off right first here talking about this idea of how does strong selection in livestock populations change the genome over time. And I almost always, whether I'm in a producer talk or a research talk, I think that it's really cool to see the power of effectively single trait selection. If we put our minds to it on a highly heritable trait like stature, we can make some pretty enormous changes in a population over a really short amount of time. If we were looking evolutionarily, right, it would take species, hundreds of thousands of years of slight changes in fitness, maybe to change as much as we've seen cattle change in stature over the last 120 or so years, right? So from this Angus cow in 1890 that looks pretty similar to the black Angus cows that are grazing in Tennessee nowadays, to a smaller but still functional steer here, or bull rather, in 1932, all the way down to these pig fat, dog sized steers that were winning international livestock shows in the 1950s and 60s. And then we switch course, of course, and can go back to something that's more moderate before we allow some introgression maybe from the Holstein breed to shoot that frame score up in the 1980s, and then selection again back to a more moderate animal that looks very, very similar to what we had 120 years ago in the sort of modern day. So all of this to say that the power of artificial selection in livestock populations, again, small effective population sizes, we can make a lot of genetic progress very quickly. We can change these phenotypes in really, really fast. But again, all of this is mediated by some sort of underlying genetic control. And my interest is figuring out, can we map the selection that's happened in really short time periods in these livestock populations that are under such strong selection, and what tools might we use to do that? And so I guess this also begs the question of, why do we care about doing this? And I really care about this for two reasons. The first is maybe a more basic biology reason. So I think that there's some understanding evolution that we can do. And in livestock, I think, are a great model organism for what a strong selection pressure exerted on a population looks like. And if we can understand how that selection changes the underlying genotypes of a complex trait, that tells us a little bit about selection on complex traits, maybe in natural populations or even in humans, right? How do we understand evolution if we can figure out how selection on complex traits works? But the other more practical application, I think, is it allows us as a breeding program, if I'm a breed association in the context of beef cattle in the United States, can we link what our breeding program is doing to some underlying biology? And with that knowledge, does that change what our selection looks like, or how we select for the traits that we do? So again, I think this can be both diagnostic for an applied sense, but also tell us something about evolution in a more basic science sense. And so just a quick reminder of how selection can impact allele frequency over time, because ultimately that's what we're talking about, right? There's some sort of a selection pressure that changes the frequency of an allele that underlies a complex trait. What does that do to the frequency of the allele over time with that consistent selection pressure? So again, drift is when we're not seeing any sort of selection on a locus. The changes in allele frequency over time are completely stochastic due to that random sampling of alleles. And this is, of course, going to be a direct function of the effective population size. So much larger effective population sizes will see fairly small amounts of allele frequency change due to drift, whereas we can get into these livestock populations and where our effective population size is in the 50 to 100 range, we can see some pretty large allele frequency fluctuations just due to the random sampling of alleles. So the second here are selective sweeps. And historically, this is what selection mapping has focused on. We want to see these large effect alleles that either new mutation pops into the population at low frequency, but it's very, very advantageous and it's quickly swept up to population, right? Very large effect sizes, very strong selective pressure on a particular locus. And that's going to create not only that quick allele frequency change for the selected locus, but it's going to change that neighboring neutral diversity around that selected site. So again, this is where we're able to go in and detect these clear signatures, whether that be stretches of homozygosity or extended linkage disequilibrium. This is sort of been what we've, as population geneticists, right, fixed our sights on are these big selective sweeps, either hard sweeps on a new mutation or soft sweeps on standard variation when a new selective pressure comes on the scene. But the fact is that in the context of a livestock breeding program, there's a few of these out there, and we certainly can detect them, and we have in a lot of different ways. But by and large, the major selective pressures in a single population like this are going to be from polygenic selection on complex traits, right? So these are traits that have hundreds or thousands of loci, of course, that underlie them, which means that effect sizes are small, but we are still expecting to see some sort of a directional frequency change with sustained intense selection on these things. The big difference between polygenic selection and a selective sweep, though, is that you're not going to see that same impact on neutral genetic diversity on either side of that causal locus, right? So what you might see that locus increase in frequency, but the strength at which selection is acting on it is so weak that it's probably not going to drag along neutral diversity in the same way that a selective sweep might have. So the challenge for us here is that if we're not able to leverage what happens around a selected locus, what tools do we have that can detect these really subtle shifts in allele frequency? And before I jump into the actual way that we approach this, I think it's helpful to say why we started with beef cattle or livestock in general, I guess. And that's that we see a lot of value in the genotypes that are being generated by ranchers and farmers in breed associations. So our handy dandy SNP chip that we've had for the last 20 or so years, not 20, even 15 or so years, has been really great, right? It's helped us make more accurate selection decisions through genomically enhanced breeding values. But really what we're interested in is figuring out if we can utilize this commercial data for basic biological discovery. And we think this is a great resource. There's millions of genotypes that have been generated in cattle. And even in some of these smaller breed associations, we're up in the hundreds of thousands of genotypes, giving us a lot of statistical power on these animals that have been born in the last couple decades, with which to detect that very, very recent selection that's gone on. And one thing that I'll throw in here before I talk about sort of results from what we've done in this work is that imputation is really the workhorse in a lot of this. So all the data that I'll present today are sequence imputed genotypes. And this is just a quick little snapshot from one of the analyses that we run trying to map this ongoing polygenic selection. And what you'll see up here is the same exact analysis run on unimputed SNP50 genotypes, whereas on the bottom is the sequence imputed. The sequence imputed version, right? And it's very, very clear that the resolution that we get allows us to better map these complex signatures of selection or whatever traits, mutations that you might be interested in, right? Having access to imputed data is at the crux of this. We'd say that there's no selection happening on the front end of chromosome 23 here, which I believe is where this is at if we were just looking at the SNP50. Whereas with sequence imputed data, we can say there's a complex signature of selection happening here, one very big LD peak here, and then a number of other sub-associations of alleles that we think might be under selection. So again, this is something that I'm also really interested in. I won't present a ton on imputation today aside from this, but we're very interested in figuring out how do we best do imputation, not only from an algorithmic standpoint, but when we build reference panels, how do we optimize this imputation problem so that this imputed data that's so valuable to us in increasing our statistical power and our resolution to detect selection or causal mutations or whatever, how do we optimize this imputation to get the best end product here? Another thing that our lab is very interested in. So I guess sort of going back to this idea of the genotypes that we get from a genetic evaluation, we have data sharing agreements with multiple breed associations, but the good thing about this and why it lends itself to the method that we develop is that we have a longitudinal distribution of genotypes, right? Certainly a concentration of genotypes towards the most recent 10 years when genomic selection comes onto the scene in the US beef industry. But we also have reached back into the semen tanks and genotyped some of these very influential and high impact sires in the breeds that we're analyzing, right? So we look back here, and there's quite a few of these sires actually dating back 40, 45 years ago that have been genotyped. And what this creates is a longitudinal distribution that we can start to look at shifts in allele frequency over time when we have this sort of time series-ish genotype data. And so the model that we use to do this is really pretty simple. We didn't go back and reinvent the wheel for this selection mapping method. Rather, we just use a normal genome-wide linear mix model, just your standard GWAS model, of course, where you've got a phenotype and you've got some sort of a phenotype typically, but what we opt to do in this sense is instead of using an actual phenotype, we're using some proxy for the generation number of an animal. And in the context of the data that I'll share today, we're using an animal's age calculated from a given date or their birth date. And so what this allows us to do is look and see if there are single SNP associations with an individual's generation number or some proxy for their generation. We call this generation proxy selection mapping, or GPSM is what I'll refer to this. And again, I think it's really elegant because it really is just looking for associations. Once we've removed that sort of familial and breed structure that exists in these data sets with the GRM, really just looking for associated variants with an animal's generation number or some proxy. And the gist here is that a significant association with generation along the genome is a locus that's responding to selection, right? There's an allele frequency that is changing over time as we look at this connection between birth date and generation number. And so the two data sets that we apply this method to are American Simmental and American Red Angus. You can read about this in a bioarchive, and it's been submitted. We're hopefully very close to publication with this, but the two data sets, again, from Simmental and American Red Angus, the two big differences, I guess, between these breeds. Simmental is an open herd book breed. So we've got individuals all the way from purebred or full blood Simmental all the way to purebred Angus that are able to register in this breed, and every cross in between. So the Sim Angus breed has become a major player in the American beef world. And as a result, we've got lots of crossbred individuals. And because of that, we've got a large amount of fairly recent introgression from the American Angus breed that's happened in Simmental, which again reflects itself as selection in the context of what we're doing here. Allele frequencies that are changing in response to that introgression of favorable alleles from Angus. Whereas Red Angus, while not a completely closed herd book, we restrict this just to purebred animals, and this allows us a little bit different look than this Simmental data set, where we're getting lots of new blood entering the herd book through introgression and crossbreeding. And so with Red Angus, again, since we're able to restrict this just to within the breed of interest, one thing that we're also concerned about when we look at this full data set, we see lots of associations across the genome, but our power is much reduced because this is not at all a normal phenotype, right? So again, the distribution has this really, really long tail. And we tried a number of different transformations to try and make this more normal, Box-Cox's, log transformations, so on and so forth. But probably the thing that was able to make this the most normal phenotype and allows us to ask a little bit different question is to restrict this just to animals that have been born in the last 10 years. So we truncate all of that distant genotyping, the super far out animals there. And what we find is that by making this appear a little bit more normal, we see a major increase in power based just on the number of associated SNPs that we identify at similar loci. And that allows us some independent associations that we're able to map when we look at just this young data set. So what this tells me in a couple ways is that we're generating some really strong selection across the genome, right? This is very much polygenic looking. This isn't one big hit on a known Mendelian loci, but in Red Angus, there's truly polygenic selection, lots of subtle shifts of allele frequency all across the genome that even when we just look within 2 generations worth of time, we're able to detect close to 100 independent loci that appear to be under some form of polygenic selection in very recent years. So a testament to the power of these livestock genomic data sets, because we have so many animals that are genotyped, we're able to pick up pretty subtle shifts in allele frequency. Most of these are, in the 1 to 2 to 3% allele frequency shifts over the last 10 years or so, right? So pretty subtle shifts in allele frequency, but we're able to detect them due to the sheer size of this data set. But in American Simmental, we see something a little bit different. So instead of truncating this into a full data set and a young data set, we look at the full data set versus just animals that are labeled as purebred Simmentals. And what we find is that in addition to being maybe a little bit lower powered here, there's just not a lot of selection that rises to this genome-wide significance level that isn't related to 2 major Mendelian loci that we know should be under selection in Simmental populations. So for folks that aren't familiar with sort of how Simmentals have changed since they were imported into the US, they start off, of course, as these really small, speckled, tan-colored imported European Simmental. And over time, what we've seen is that they lose that color pattern and they lose that color-sidedness. And sure enough, the 2 loci that are under the very strongest selection in purebred Simmental over the sampled data set from, the 1960s, I guess, is when registration number 1, and he's genotyped in a part of our data set, all the way up to modern day animals that look very similar to an Angus animal. So this is the heaviest registration bull in the I guess early 2020s, and he's purebred Simmental by pedigree, but looks very, very similar to an American Angus, right? And sure enough, we've selected against color-sidedness in this KIT locus that controls it. We've seen a large decrease in the allele frequency there, and then as well as against this PMEL ERBB3 locus, which controls this sort of tan or red coloring of the imported European Simmental. And again, these are 2 things that make our modern Simmental appear more and more like a purebred Angus would. The big difference, though, is when we look at the full data set. So we get a little increase in power due to more animals, but really the important thing is that we see almost 5 times as many independent associations within this full data set. What that tells me is that there's been a lot of selection, or allele frequency change rather, that's been driven by that introgression of American Angus into this Simmental population. So when we look at the whole population, we detect more selection, whereas it's really limited to a couple of major Mendelian loci if we're just looking in the purebred breed. So lots and lots of the genetic improvement, I would guess, from American Simmental is derived from this introgression of American Angus into the population. So I won't get too into the weeds about individual loci, but I guess overall what we find that we're actually selecting for beyond in Simmental sort of coat color and breed characteristics, we're selecting for increased growth, which is exactly what we'd expect for, exactly what genetic trends over time would suggest, that many of these signatures of selection that GPSM picks up are underlying major growth QTL across the genome Lots of novel signatures that we don't know what's going on, but many of these are underneath annotated QTL that allow us to say pretty confidently that we're selecting for increased growth. And especially in Red Angus, we're selecting for a number of different maternal traits. Again, using sort of curated QTL, we're able to say with some pretty high confidence, and we run some QTL enrichment analyses, and we find that by and large, the QTL that underlie these are very involved in growth, maternal function, as well as a number of immune genes that look like they're under selection. So I know that we're rarely, especially in the beef cattle industry, outright selecting for immune function or increased health traits, but in a lot of ways, this is probably just being drug along by selection for increased performance, healthier cattle, being more likely to perform well in whatever setting they're in, right? And that's just dragging along selection for some of these immune genes and immune functions in these animals. So I guess an interesting takeaway, something that we'd certainly like to dig into a little bit more. We don't have a great understanding, of course, of how immune function is genetically controlled in cattle, but we'd be very interested in seeing how selection for a variety of traits is actually impacting the immune system's functionality from a genetic perspective. So I want to dig into a couple of these signatures and just show, I think, the power and interest in why this GPSM method is really exciting for us. So in both of our data sets, one of the largest and most significant peaks when we look across the genome is here on the end of chromosome 23, significant in both of these data sets. But when we dig into it, it looks like there's a complex signature of balancing selection that's going on. And I say that for a couple of reasons. First, you'll notice that the signature here, when we zoom into the first 2 megabases of the chromosome on the Red Angus data set versus the Simmental data set, the major peak here in Red Angus is different than the most significant little sub peak over here in Simmental by, again, an order of magnitude of negative log 10 p-values almost on what you see over here. But the interesting thing is that when we looked at the actual allele frequency trajectories at the lead loci, so over here in Red Angus and over here in Simmental, the Simmental signature looks a lot more similar to what we would expect under polygenic selection. So that subtle shift in allele frequencies over 4 or 5 generations. Whereas in Red Angus, we see this oscillation of allele frequencies up and down as we look over time. And to me, these oscillating allele frequencies around an intermediate value, that sort of screams balancing selection. So we run a number of tests for balancing selection to GMAS D, so on and so forth. And this region just absolutely lights up as an area of the genome that appears to be under some form of balancing selection. This intermediate allele frequency appears to be the preferred the preferred frequency here. So again, an exciting way for us to not only detect this directional shift in allele frequency, but also some of these more complex signatures as a form, I think, not necessarily of, it doesn't immediately tell us anything, right? But I think that this allows us to dig into some of these things, build hypotheses about why there might be balancing selection around this gene that's been implicated in fertility, immune function, in a number of different things. So an exciting way, I think, for GPSM as a tool, not necessarily to rely on any sort of selection on a phenotype, but phenotypically, agnostically, give us an ability to dig into some of these loci in some more depth. So the last thing that I'll point out with regards to this method is that it shows minimal overlap with different selective sweep measures. So we run a haplotype-based method for mapping selective sweeps and a composite metric that's much more site frequency spectrum derived in nature. What we find is that there's minimal overlap. There's some, but not a ton of these GPSM loci that end up being genome-wide significant for these other sweep mapping methods. And then when we dig into them more, you'll notice, of course, that these GPSM loci, which we're saying are indicative of polygenic selection, we don't see the same sort of extended linkage disequilibrium here than we do in the two different sweep methods that we utilize, right? So again, these are subtle shifts over fairly short periods of time that don't generate these big signatures of selection that, again, when we look at the lead alleles, I guess, in these two different sweep mapping methods, these are pretty flat allele frequency trajectories, suggesting that the selection that they're picking up on is distant in the past, whether that's breed formation or early genetic improvement, right? Whereas the changes that we're picking up with GPSM have to have occurred during the sampled time of alleles that we're looking at, right? So if there's selection that happens before we get a sampled genotype that we analyze with GPSM, we're not going to be able to pick up that allele frequency change unless it happens within that set of genotypes that we're looking at. And so with that, I think that this suggests to me anyways that selective sweeps don't tell us an awful lot about selection on complex traits, but I think that GPSM is an intriguing way for us to dig into complex trait selection, and it pairs well with sweep mapping methods. They're very complementary. And the last thing that I'll talk about along this line is this tug of war between artificial selection and local adaptation. So I talked there for a second about artificial selection. Farmers make breeding decisions that make them more profitable. That drives underlying allele frequency changes that make the farmers more profitable. But occasionally, our hypothesis is that this might be at the expense of local adaptation. So animals that are consistently bred in a single location, we would expect them to, if they're selected based on performance, there's some level of genotype by environment interactions there. We'll see that underlying locally adaptive alleles will be different between populations that are in different environments, right? So there's this sort of tug of war between evolution in a very classical sense and this artificial selection that producers are making. And so we utilize a similar method to GPSM to try and see if we can map these allele frequency differences, both from a continuous environmental variable and also utilizing these environmental zones of the United States. So we take these big commercial genomic data sets from breed associations, and we're able to tie individuals to their GPS locations and estimate what the climate is there and how that relates to other climates across the US, or using sort of long-term climate data to classify them on a continuous sort of metric. You can read about this in a PLOS genetics paper that we came out about a year and a half ago. But again, this is just a normal GWAS, but instead of using some sort of a trait or like GPSM using age, we're just using one of these continuous variables or a sort of case control phenotype on whether or not an animal resides in one of these environmental zones. So what we find when we run these scans, this is an example Manhattan plot from the Red Angus data set that is a multivariate GWAS for temperature, precipitation, and elevation at each of these locations. You can see the geographic distribution of the Red Angus breed across the US and some nice gradients in all three of these things as we look across the country. And what we find is that there appear to be a number of loci that have significant associations with these continuous environmental variables. These underlined ones have been identified in local adaptation studies in humans, and other examples have been identified in livestock or model organism adaptation studies. So again, this is much more in the hypothesis generation stage, right? We're pointing out some of these things that are associated with environments, but the actual functional validation of this is of course much more difficult. But again, I think a convincing sort of first step for us to say that these allelic associations with environment exist, and they also exist when we utilize this sort of case control ecoregion way of looking at things, right? Significant associations across the genome, but we're still maybe statistically underpowered, or there's just not a ton of these that are actually out there that remain significantly associated with an animal's environment. Of course, the one consistent locus that appears to be environmentally associated is this one on chromosome 23. Of course, a perfect overlap with that candidate balancing selection mutation that appears to be oscillating in allele frequency at intermediate frequencies in Red Angus in the GPSM analysis. And this is a sort of classic case where balancing selection might be at play. This local adaptation or changing environments or whatever that might be, right? An interesting thing for us to probe into in the future, but I don't have a great explanation for that at the moment. The last thing that I'll mention here is that when we take these nearby genes from the local adaptation or environmental association GWASs, and we throw them into a pathway enrichment analysis, looking at the genes themselves, completely no overlap, right? We're getting a completely different set of genes in Red Angus and in Simmental and in Gelbvieh. But when we throw these at a pathway enrichment analysis, we see that there's a number of these different KEGG pathways that appear to be shared between the local adaptation hits in Red Angus, Simmental, and Gelbvieh, almost all of them having to do with neural development and function. So as it would make sense that some sort of neural function is likely driving these local adaptation differences, an exciting first result that we haven't dug into a whole lot more in depth, but an interesting way for us to look at these hits that might not match up when we look individually at the GWASs, but there's some sort of conserved biology that's likely governing local adaptation in these populations. And finally, the last thing is that when we squish these two methods together and we ask the question, are there alleles that are changing in frequency over time in a region-specific manner? We find some, but the unencouraging thing is that in almost all cases, these region-specific allele frequency differences, which may or may not be local adaptation, are actually shifting back towards the population mean. So there's really no differentiation that is ongoing with regards to local adaptation. But what we might be seeing is previous local adaptation, these adaptive differences that are actually regressing back to the population average. So the artificial insemination that we've done that's generated enormous amounts of genetic progress might be at the expense of some of these locally adaptive alleles actually leaving the population. So with that, I think that this is justification for the U.S. beef industry probing this local adaptation or figuring out how we might deliver sort of environmentally informed predictions to our producers. So I think that this is an interesting first step in this area, but something that we need to be conscientious of is beef cattle, as we all know, still that last extensively raised species out, subject to all of the environmental stressors that exist. So something that we're very conscientious of in looking at ways that we might actually deliver some sort of an environmentally informed breeding values in the future. And so I spent a lot of time on those first few slides, and I'll fly through these last couple and leave some time for questions for us here. But the next question that our lab really has is, how does heterosis work? And I have to give credit where credit is due. This is a project that we got funded last year, and it's really a result of a famous Twitter account, the Cats of Animal Breeding, if anybody's on Twitter. This is a really entertaining one, but for a long time, Cats of Animal Breeding was talking about hashtag solving heterosis. And that got me thinking about what solving heterosis might look like. And so we've sort of tried to bite this off, and we're really excited. I don't have a ton of data to share as we've just started generating some of this, but I think the prospect of utilizing a knowledge of how heterosis works sort of under the hood is an exciting way for us as animal breeders to get a little bit closer towards making mating decisions in a more precise way to take advantage of the underlying mechanism behind heterosis. So the corn folks know all about heterosis. They're very good at it, but U.S. beef producers are not nearly as good at it as they are. So even though the traits that we struggle the most in a commercial cow-calf sense to make genetic progress on, so maternal ability, fertility, health, immune function, these are the things that are the lowest heritability, but of course show the highest levels of heterosis. But less than 50% of our U.S. beef herds are utilizing crossbreeding in a meaningful way. So I've made this sort of my extension and research crusade to help our producers understand what's going on underneath the hood in heterosis and how they can take advantage of that in their herds to really to not leave free money, free performance on the table by crossbreeding. And so we're all conscientious that heterosis happens at the level of the organism. So whether that's added weaned calf pounds, if that's additional females that are getting pregnant each year, heterosis happens at the level of the animal. But we're also it has to happen at the level of these intermediate molecular phenotypes, and that's where this problem and this project in our lab fits into the unconventional phenotype realm, right? So what we're interested in is saying, okay, we know that heterosis happens at the level of the animal. Does it also happen at the level of gene expression? And we all know that gene expression is this really complex network, right? It's tightly regulated, but can we identify gene expression heterosis? And will that allow us to understand a little bit about how these complex traits are affected by what happens under the hood of heterosis? And so as much as we'd really love to go out and make a big resource population of hundreds of crossbred animals and purebred animals, the USDA really doesn't like to fund these sort of super long-term, expensive processes where we're making a bunch of animals. So what we opted to do is control the situation as much as possible. It seems like you sort of sneeze at an animal and you can change gene expression, right? But our goal is to reduce the environmental variation in gene expression. And so we've decided to, for this particular research question, move into the lab and generate some in vitro embryos and study heterosis at that level. We can do it on a lot bigger numbers, a lot cheaper, and in an environment that's much more controlled, so that we're able to pick up these maybe more subtle gene expression differences between purebred embryos and crossbred embryos. So we focus, of course, on this late stage blastocyst. So as long as an embryo can survive in vitro, the embryonic genome is activated. It's its own organism. We've got some cell type differentiation, of course, from the intercell mast and the trophectoderm. So we think that this is an exciting model for us to look at large data sets of animals in a fairly cheap manner. And so what the study design here is that we're making a sort of diallele cross, leveraging abattoir-derived ovaries from Angus and Brahman cattle, multiple different AI sires. So we're able to sample across these two breeds on the sire side, and some variation, of course, from the dams that we're able to go and collect ovaries from, and create somewhere in the neighborhood of 200 to 250 mature blastocysts, about 50 from each purebred Angus and purebred Brahman, and these reciprocal F1s. So we think that this is a really powerful way for us to go in and start understanding, are there differences in expression in these F1 individuals? We can get at parent of origin specific expression, imprinting, in some of these, the role that epistasis might play in these gene expression phenotypes. We're also going through and sequencing each of these sires in samples from each of these dams, about 5x on the dam side and 10x on the sire side, which will allow us to pool these embryos in their development, assign parentage to each of them, and then hopefully do imputation with relatively high accuracy on these transcriptomes. So what that eventually, of course, will allow us to do is map expression associated QTL, something that hasn't really been done a ton at this particular stage of development that we're very excited about. And I'll sort of breeze through some of this stuff as I'm getting close on time. I'd like to leave some time for questions in respect to y'all's hour-long seminar here. But again, of course, our big question is can we identify misexpression? So expression that's either higher or lower of certain genes in this F1 offspring, and then see how that fits within our gene expression networks. These things are super tightly regulated, but we know that there's certain genes that drive expression of a number of others, right? And knowledge of that, we feel, is really important to understanding the molecular basis of heterosis. Like I said, we're also very interested in using the imputed transcriptomes to go back and actually map additive and non-additive expression phenotypes. And then with the advent, of course, of single cell RNA sequencing, we're very interested in seeing if there's tissue-specific expression differences between the purebred and F1 embryos at this very early stage in development. So something that we're also going to explore with our 10x capabilities here at UT. And then trying to make some guesses about what the downstream impacts of these things might be. So are there developmental differences in changes in gene expression, say in this inner cell mass or in this trophectoderm that'll go on to become the placenta, that are experienced downstream that we see when we look at maybe adult populations of purebred or F1 individuals. And so in wrapping up here really quickly, the other big thing that our lab is interested in is cow efficiency. And I'll skip through some of this, but the big issue, of course, is that phenotyping the things that we want to select on is tricky, right? So we're very good in the U.S. beef industry at measuring birth weights and weaning weights, right? Our cost of entry here is a hand scale and a lack of fear of this mother coming after us. Again, having a platform scale, you can collect weaning weight data. But as we get to some of these more difficult to measure or more expensive traits to measure, think carcass traits or feed efficiency, the number of phenotypes that we have are much lower. And as a result, our genetic evaluations are not to the same level of accuracy as things like birth weight or weaning weight might be. But then as we sort of move into the even harder and more expensive traits to measure, so of course, greenhouse gas emissions, something that the folks at Guelph, y'all are some of the world leaders in doing this in the dairy world. But this is really, really expensive for us to measure, especially on beef cattle that we're seeing maybe once a week are coming to a common area versus a couple or 3 times per day, like a dairy cow. These things get really hard and expensive to measure. And then as we try and get deeper and deeper into the biology that underlies these traits, right, towards that sort of molecular or biological mechanism, measuring that is really expensive and hard for us to do, even though the knowledge of these would be very, very important to our breeding goals and our genetic evaluations. And so, again, the challenge here, whether it's in the state of Tennessee or whether it's in the state of Texas, where my wife's family ranches, we're just not seeing these cows as frequently. We have to be able to observe these phenotypes out in their natural environment. And frankly, that's just it's not practical for us to utilize a lot of the same technologies that might work in a dairy or that might work in a feedlot. Much more difficult to implement when we've got these big rolling hills or mountains in Tennessee, or we've got cows that are stocked, one animal to 30 or 40 acres, like we would be out in West Texas. So what we're leaning on here at UT is figuring out which technologies we can borrow from our friends in the dairy world, the ones we can borrow from folks in the feedlot world, and apply these to cow-calf systems. So we've been playing around with these accelerometer ear tags. We've got green feed systems that have just gone online, so we're measuring not only these phenotypes like greenhouse gas emissions, but also maybe some more direct measures of animal metabolism that we're able to triangulate from that green feed data, and a number of different things that our precision livestock farming group here at UT is working on and trying to tie genetics into this picture. So both in a phenotyping sense and also in a precision management sense down the road. So the big trait that I'm really interested in is pasture-based efficiency, but this is a really, really noisy one and one that's tricky for us to measure. We can go out and feed cattle in sort of a feed efficiency testing setting, but we know that that's not perfectly associated with what happens on pasture. So there's these metabolism differences, of course, between cows. Some cows are just better at using energy and in devoting it towards raising a calf than others. But there's also this idea of variation in grazing behavior. There's weather and climate that plays into this. So how a cow sheds off her winter coat early on in the season might impact how likely she is to graze, which again is going to impact something like feed intake on grass. And then, of course, seasonal variation in forage. There's some sort of G/E here that's likely happening. So we see this as a major problem for us, and something that we have producers that are very interested in making breeding decisions that are focused on identifying these more efficient cows. And so we just recently got a project funded through the Foundation for Food and Agricultural Research here in the States to combine precision phenotyping. So we partnered with CLOC, incorporated on not only methane measurement devices, but also we've been working with them on some water intake sensors that pair with their real-time weight gain scales. So figuring out in these forage-based beef cows, what's happening in real time. How are they gaining, losing weight? What's their water intake doing? What's their respiration, methane emission, all of these pieces. And then combining that with activity sensors, so figuring out grazing behavior, relative temperature during times of heat stress, as well as reproductive phenotypes. And then finally tying all of that together, not only with genotypes, but also with these molecular intermediate phenotypes. We've been playing around with this 3' RNA-seq quite a bit in the lab. We're really excited because we've gotten our costs down to $30 or $40 a sample to get a full gene expression profile on these animals, as well as metabolites and collaborations with some folks that do microbiome research. Again, get a comprehensive picture of what we might use to form these intermediate cow efficiency phenotypes using these biomarkers that we identify. So we're in the process, of course, of starting to collect this data, but this is going to be really focused on modeling or creating models, right, that are able to identify important molecular features, whether that's a metabolite, whether that's an expressed gene, and determine what our repeatability over seasons or contexts or certain environments are. And ultimately, our goal is to see if we might be able to use some of these molecular intermediates as phenotypic stand-ins, right? Figure out if we can piece together using a number of metabolites or a number of cheaply measured genes that appear to be expressed in a sort of reliable way over time as indicator phenotypes for these hard or expensive to measure traits. And so I'll skip through this and leave you with a couple of things before I take a couple of questions here at the end. The first is if you're involved in any way in precision livestock farming, we're hosting here at the University of Tennessee the National PLF Conference. It should be a really good mix of folks from across the states and Canada coming down and we'll really across species, across disciplines, but all the folks that are integrating precision livestock farming into their research. We'd invite you to join us in May in Knoxville. It should be a really great conference. I'm happy to answer questions about that. And then I'm also excited to join you all north of the border this July for the annual symposium of the Beef Improvement Federation. That's going to be in Calgary right before Stampede starts in July. So we're really, really excited to take the Beef Improvement Federation north of the border. It'll be my first trip to Canada, and there's a really great group of speakers. Some folks from Guelph I know will be over there. But our focus here is on precision breeding and all the different things that entails for beef improvement in the US and Canada. So with that, I'll leave you with some takeaways and thanks from our funders and cooperating breed associations. And I'm happy to take any questions. I know that I breezed through a lot there. So if I can clarify or give any more detail, I'm happy to do that.
Great. Thank you, Troy, for the fantastic talk. So yes, I'd like to open the floor to people for any questions. Feel free to raise your hands. Christine?
Thanks for the great talk, Troy. I'm wondering what we have to do to get some of those beef phenotypes that you collected on feed efficiency and methane emissions. If you're interested in collaborating or able to, what's the story there?
Yeah, no, we would certainly be interested in collaborating. We're just, like I said, these things have just gone online. I spent the first year and a half at Tennessee convincing people we needed to buy these devices. So we're in the early stages of phenotypic collection, but we're going to be hitting hard this next year. So we should certainly talk about how we might be able to borrow some information and ideas across the beef and dairy space. Because again, I really do look up to to your guys' group and what you're doing in dairy. And so if we can replicate some of that or learn from some of your challenges, we'd really like to do that. And if you think there's applications in the beef side that we're not thinking of, we'd certainly be interested in hearing your thoughts on that too.
For sure. We're actually in the middle of a process right now, applying for a new large-scale grant, and we've got partners in Ireland and Australia who are going to be working on the beef side as well. Our focus is, of course, and remains on dairy, but
sure,
this new project is really focusing on if there are any places that we can make 1 + 1 = more than 2 with regards to genetic evaluations and what we learn. So I'll be in touch anyway. This is probably not the platform for that, but great talk as always, Troy. Very enthusiastic and just really impressive what you guys are doing down there. That's
awesome.
Really
Thanks,
great.
Christine. Yeah, stay in touch about that.
Absolutely. Ciao.
Thanks, Christine. Flavio.
Yeah, can you hear me?
Yep.
Yeah. Okay. Troy, wow, excellent presentation. Really, really informative. Thanks for doing that. I have a question. When you presented the first study on the association with the age, right? The longitudinal one. I wonder why you didn't think to fit in the model inbreeding of the animals, right? Because along with the selection process, you're going to have a drift because of the effective population size that is changing, right? So I don't know if only the relationship matrix, the genomic relationship matrix would adjust for that, right?
Yeah.
So maybe I was thinking, why not put as a regression coefficient there the inbreeding of each animal to remove that association of the alleles with the year, right? That could be due to the inbreeding, therefore coming from more drift than anything else.
Sure, that's a really good point, and no, we didn't explicitly fit anything past the GRM from an inbreeding perspective. I'm trying to work through how that would look. I didn't include any information here, but we did a ton of simulations. I worked during part of my PhD with John Hickey's group on some simulations for this, and certainly even with really small effective population sizes. We're still able with this method to distinguish from those really small allele frequency shifts from drift even. So across simulation space, it works, but we hadn't even thought to fit an explicit inbreeding coefficient, but that's a great idea.
Okay. May I ask another question? I don't know.
Yeah, work away, Flavio.
Yeah, okay. With respect to the other study, the GWAS with the environment being the phenotype.
Sure.
Again, I know that you included the genomic relationship matrix in the model, but how was the distribution of the sires across the environment? Have you tried a scenario where you would force to have a very good distribution of the sires, right? Across the scenarios to compare with just taking whatever the data you have and fit the relationship. Again, I'm thinking about the stratification that could be created by the more intensive use of certain sires in certain environments.
Mm-hmm.
So that kind of association of the alleles with the environment, but just due to the use of more intense use of certain sires.
Yeah, that's another really good question, and something that we've looked a little bit at, and there's certainly some use of sires across these different environments, but I would say it's much less in U.S. beef populations than in dairy populations, right? Artificial insemination, even in these seed stock breeds, is much lower. So we're not seeing a ton of sires that would be overrepresented, or there's not a ton of genetic connectedness across these regions, or certainly not as much as you would expect to see in a dairy population, right? But that's another thing that we could definitely delve into in a little bit more detail, figuring out how overrepresentation of sires in certain certain regions might be driving those frequencies past what gets handled in the GRM.
Yeah, no, I think that would be another additional information that you could dig in. Yeah. Okay. Thank you so much for the presentation. Really good.
Yeah. Thanks.
Thanks.
Thanks, Flavio. Alan?
I've been told by beef cattle people, mainly Ron Lemonager, when he was doing the NRC model for beef cattle, that the greater the milk production potential, the greater your maintenance requirements all year round.
Yep.
And that some of that may be due to liver metabolism. And I'm doing some modeling of heat stress in sows, but the liver produces like a third of the heat in a lactating sow. But is some of your cow efficiency going to be that you match the animal's milk production potential to the environment, so that if you put too high a producing animal in the environment where the feedstuffs are not adequate, you'll still have the maintenance requirement, but you won't have the milk production? But
right. No, I think that's spot on, and that's something that we've in the beef industry. I think we've done an adequate job of informing our producers of that there's a cost to milk, right? And again, so much of that is matching, like you said, forage requirements with that genetic potential for milk or that genetic potential for growth. And I think a big piece of that is that we're going to figure out or that we're interested in, right? Is can we observe these metabolism changes or the underlying biological changes that are associated with those times where lactation overlaps maybe with either a heightened or lower quality or abundance of forage. So I think that's certainly going to be a huge part of this holistic picture of cow efficiency, and something that we know about from a quantitative genetic standpoint and from a beef cattle management standpoint, but that we've had a trouble of untangling the biology underneath that. So we're certainly interested in figuring out how milk production is tied into all of these resource uses in metabolism. So that's a great question as well.
Great, thank you, Alan. And since we're a few minutes over time now, I think we'll probably close it up there. But just wanted to thank you again, Troy, for the great talk. Great way to finish the semester. And thank you to everyone for joining us throughout the semester. We will be back in the new year with 2 new seminar hosts. Samla and Kristen will be taking over in the new year, so me and Ivan will be finishing, but it was a pleasure to do the seminars for the last 2 years, and thank you, Troy, for the great talk to end my reign as being in charge of these. So thanks again.
Sure. Thanks again for having me, Colin. Y'all take care.
Cheers.